Today we are going to see how we can identify the clones from a gene library, or you can identify or screen your recombinant cells which are carrying your gene of interest. So we can have either your direct method or indirect method. In direct method, it is where the vector itself is altered in such a way that the insertion of your gene of interest will give you some kind of your changes that could be depicted. For example, you have insertional inactivation. So insertional inactivation, two types are there. Like you can have direct antibiotic resistant gene inactivation, where within the antibiotic resistant gene, the gene of interest is inserted. So depending upon whether your recombinant cell is or the cell which you are checking, whether that cell is sensitive or resistant, you can select your recombinant cell. Similarly, you have another way, that is your blue-white screening method, where your laxate gene is being used. So those are your direct methods which are definitely preferred, but sometimes we are not able to use those methods. So we have to go for your indirect method. So in indirect method, you have two types. Like the main two types, there are many methods, but main two types are your nucleic acid hybridization, and the similar assay is there, that is Plaque hybridization method, and where we would be requiring to have at least some knowledge of your gene of interest, because here we will be requiring the probes. So probes which are going to be complementary to your gene of interest. So that is the reason why prior knowledge is definitely essential for that. The next method is your functional screening. So in functional screening, usually is used for your expression vectors, where actually the gene is going to produce your translational product, that is your proteins are there. So we will be using some method to identify, to screen whether your protein of interest is being produced by your recombinant cells or not. So that can be done with the help of the antibodies. So let us move ahead and understand the first method, that is the nucleic acid hybridization. So this particular method is used for the detection of individual clones in a library, or you can also have your recombinant cells. Okay, so by employing your nucleic acid hybridization using your a particular probe. So probes are nothing but they are small chemical synthesized label oligonucleotides. Okay, so which are going to be complementary to the sequences in individual cells, or you can also have your phages. Sometimes if you are using viruses for storing your gene library, or for even your recombinant clones, so for that case also you will be able to use your phage hybridization. So it will always these probes are going to be very very important, as we have already discussed that prior knowledge of the gene is required, then only we can design a probe. So if probes are available, then this is the best method to go for. So the probe must share at least a part of the sequence of the cloned gene. So if the gene itself is not available, so it can be derived, for example, from some known protein sequences. Like you have to go for first you'll have your protein sequence. From protein sequence you are going to find out the gene sequence, and from the gene sequence then you would be that if you have the gene sequence, it would be helpful in producing a particular probe. Okay, so that is how your in this thing can be used. So even base substitutions and or identical at different positions, that kind of your mixture of your oligonucleotide probes can also be produced. So the first step of your hybridization screening experiment involves is your transfer of the DNA. So be it from the plaque or from your colonies. Okay, so these DNA has to be transferred onto the nylon or your nitrocellulose membrane. So simply your bacteria or your recombinant host cells which are there. Okay, so they are going to be they are going to be lysed, so that the DNA will be released, and then this DNA is they would be lysed, and this DNA is transferred onto the membrane. After that, that membrane or whatever your DNA is there, it has to be denatured using alkali treatment. Okay, so that you can have your single strands. Now, why your single strands are required? Because your probes can go and hybridize only to your single stranded DNA. So that is the reason why this alkali treatment is very important. Then these DNA which are there, they can be also bound to the membrane using your heat treatment or your UV irradiation, so that whenever we are giving wash, the DNA should not get washed away. They should remain bound. Okay, so the membrane is then immersed into a solution containing your nucleic acid probes. Okay, so these probes are radio labeled. So when we are incubating, and if your mixture of your DNA contains your DNA of interest, which is for which the probe is complementary to, then the hybridization will take place. So after hybridization, the membrane is washed so that whatever your unhybridized probes are there, they are going to be removed, and then you can visualize the probes using your autoradiography. So this is the way how you can see this. So you have your nitrocellulose membrane. This is the master plate, okay, which is going to carry the colonies of your bacteria, which is expected to carry your recombinant plasmids. So your nitrocellulose membrane has to be placed on this. So what will happen is the colonies from this petri plate will get transferred onto this nitrocellulose membrane. So once you get this thing, then the bacteria are going to be lysed. So when they are lysed, the DNA will come out, and the DNA is going to be denatured using your alkali treatment. Okay, so after the alkali treatment, yes, they are also going to be given treatment with UV radiation or heat treatment, so that they get bound to your membrane. Then you will allow it to hybridize with your radioactively labeled DNA probes. So you can see, and then you can expose it to your photographic film to see whether these spots are being created or not. Okay, the positioning. Okay, so wherever these spots you are getting, the positioning of that can be then again compared with this master plate of the colony. So you can see over here, these are the positions where your spots are being identified. So those positions, so this is similar to your replica plating. Okay, you already know the replica plating technique. So similarly, you have to just compare this position with this position, and then you can identify which are your cells which are producing these whatever your clones are there, or your specific DNA, your recombinant DNA is being carried by which of the cells, that can be identified by your this hybridization technique. Okay, so similarly, you have this Plaque hybridization method, which is similar thing. It is only that instead of your bacteria, it would be your plaques that would be present. The rest of the things will remain the same. So Plaque hybridization is also known as your plaque lift. So it was developed by Benton and Davis way in 1977. So you have this filter lift method, which has been used for this. So the procedure is very successful, for isolation of recombinant phages. So the same, it is your nucleic acid hybridization only. So because instead of your bacteria, we are using plaques, hence it is called as your plaque hybridization. So the nitrocellulose filter is applied to the upper surface of your agar plates, so that your direct contact between your plaque and the filter is there, and then the same thing is going to be again happening. So you can see you have the plaques. So plaques are nothing but when viruses are bacteriophages. So for example, your lambda-phages are growing onto your lawn of bacteria. They will form patches, okay, where these bacteria are going to be lysed. So those are your phages which are rich on these areas. These phages are going to be rich in the viruses. So nitrocellulose membrane is being placed, and then yes, they are being lysed so that the DNA is released and then denatured. They are bound to your these filters, and then the same thing you will go for your hybridization with your labeled probes, and then autoradiography. Okay, So this is about your Plaque and your colony hybridization methods. Okay, so colony hybridization and Plaque hybridization, these are the two types of your nucleic acid hybridization methods. Okay, the concept is same, only in colony hybridization it is your bacteria, and in Plaque hybridization it is your plaques that are transferred onto your filters, that is your nitrocellulose membrane. Now coming on to the functional screening. So functional screening here, we are talking about the activity, the function of your desired gene. So what is the function of your desired gene? To produce your proteins. Okay. So here, in this case, definitely either we are screening your expression libraries or we are screening your recombinant cells. So you can find in that case. So functional expression of cloned DNA fragments are there. Functional gene products and hence the desired clones can be detected either by antibodies. So your immunological screening, that is what is there, or you can use some kind of ligands. If your proteins are having some specific ligands for binding, so that ligands can also be used to recognize the encoded proteins. Okay, so the bioactivity of the gene product. Okay, whatever your proteins are there, the bioactivity is going to be exploited in this particular method. So you have this master plate. Okay, so from the master plate again you have it has been transferred onto your nitrocellulose membrane. So uh, then the cells are lysed so that now the proteins, okay, so in this case the proteins are going to be bound onto your surface. So then you will be giving treatment. So in case of your colony hybridization or your nucleic acid hybridization, you will giving a treatment with your, or you were allowing the hybridization with your probes. In this case it is your antibodies that you will be allowing it to bind to. So first you can give treatment with primary antibodies, which will be directly going and binding your protein of interest. So whichever bacterial cell is producing your protein of interest, so against that for which your primary antibodies have been added, so those antibodies will go and bind. Once it has gone and bind to that, you will be giving a wash so that whatever the unbound ones antibodies are there, they would be um, washed off, and then you add your secondary antibodies. So secondary antibodies are those antibodies which are going and binding to your, or which are specific to your primary antibodies. Okay, so they will go and bind to your primary antibodies, and these secondary antibodies are going to be your enzyme labeled. Okay, so this is a very much similar technique as your ELISA. So enzyme labeled, when you give an add substrate, so these substrates are your chromogenic substrates, that means it will produce color. Okay, some or the other color is going to be there, so that color production, so you can, is going to be considered as your positive signal. You will save spots at that particular point again. So you can see here, because this particular place your spot is there, you can correspond it to your master plate where your colony is there, and so you can take those colonies and subculture it onto your or isolate it or grow it in your liquid medium, so that you have those cells, so you are able to screen out those cells which are actually going to carry your protein of interest. Okay, so that is how. So the procedure is very much similar as we have already discussed. So the membrane is treated with covalently attached protein and immersed in a solution with antibodies. So these antibodies will go and bind to the epitope. Okay, so that is specific to your protein of interest. So this is the same thing that has been shown. Okay, so bacteria get transferred onto, so this can also be done for your phage libraries itself. So instead of your bacteria, if we replace it with phages, that can also work out. So just one thing we have to remember over here, that here it is your antibodies that are being used for detecting the your proteins. So similar type of your functional things are having, there are two other methods also which can help us in identifying the protein of interest, that is your HRT and HART, which we will see next time.